any kind of stimulation, and as a result, mercury from fillings produces the majority of human exposure to mercury. The International Academy of Oral Medicine and Toxicology is extremely concerned about the anecdotal claims of safety by manufacturers and dental trade associations that are at variance with the published peer-reviewed scientific evidence to the contrary. The precautionary principle requires action once the possibility of harm exists. It does not require proof beyond a shadow of a doubt that in the case of heavy metal and xenobiotic exposure is both nearly impossible and unnecessary, in our opinion. What you're seeing is mercury vapor coming from toxicology, set out to determine the amount of mercury that was coming off fillings. And here's the graph, showing substantial quantities of mercury were measured coming off fillings. And then we estimated the total dose. And then we began animal experiments. Bacteria that were antibiotic resistant cropped up in the intestines within 2 weeks of receiving these mercury-leaking fillings. Further studies have found damage to the ADP ribosylation of brain neuron proteins. In response to the controversy, and at the request of the Federation of Experimental Scientists and Biologists, Drs. Fritzloh-Scheider and Murray-Vimy, that should be of concern to anyone wanting to have healthy children, because mercury is highly damaging to fetuses.
How mercury causes brain neuron degeneration. Shown here is the neurite of a live neuron isolated from snail brain tissue, displaying linear growth due to growth cone activity. It is important to note that growth cones in all animal species, ranging from snails to humans, have identical structural and behavioral characteristics and use proteins of virtually identical composition. In this experiment, neurons also isolated from snail brain tissue were grown in culture for several days, after which very low concentrations of mercury were added to the culture medium for 20 minutes. Over the next 30 minutes, the neurite membrane underwent rapid degeneration, leaving behind the denuded neurofibrils seen here. In contrast, other heavy metals added at this same concentration, such as aluminum, lead, cadmium, and manganese, did not produce this effect. Shown here is a neurite growth cone stained specifically for tubulin and actin, before and after mercury exposure. Note that the mercury has caused disintegration of tubulin microtubule structure. These new findings reveal important visual evidence as to how mercury causes neurodegeneration. More importantly, this study provides the first direct evidence that low-level mercury exposure is indeed a precipitating factor that can initiate this neurodegenerative process within the brain.